filtration begins at the glomerulus. The glomerulus has a very characteristic wall structure in those capillaries. It has 3 layers. Each layer does a specific role in the filtration process. The first layer, so on the inside lining of the capillary, that first layer has a whole bunch of pores. They're called fenestrae. And so we say that the capillaries are fenestrated. Glomerular capillaries are fenestrated. It just means they have large pores, and that allows water to pass through pretty easily, and things that are dissolved in the water can pass through. What can't pass through are large objects like blood cells. So red blood cells, white blood cells, all right, we don't want those to get flushed out with the urine. We want to hold them back. Those are important components of blood. So those get held back. They do not pass through the pores. Next layer we have is the glomerular basement membrane. This is made of a lot of connective tissue, lots of collagen is present here. And this layer, it really helps to restrict the rate of fluid flow. So we don't want the blood to be flowing, we don't want the filtration has to happen at a set speed. It can't be too fast or it might not do a good job. So this glomerular basement membrane controls to some extent the rate that fluid is flowing across, and it also provides a barrier for plasma proteins. Plasma proteins, again, like proteins needed for clotting, for example, we don't want to just flush those out with the urine. We want to keep those back and save them in the blood. So that layer helps with that job. Finally, the last layer right here, still talking inside of the glomerulus, this last layer has special cells called podocytes. Podocytes, so podo, this means feet. They have long processes like feet, really extended feet, and what those podocytes do is they sort of lie adjacent to each other. Those long processes are adjacent to each other, and they form narrow slits. Those slits are bridged across by connections. So there are links between the processes, and this together forms like a really fine mesh network. So this is it's called a slit diaphragm. There are lots of slits, and it's a diaphragm. It's something that that the filtrate passes through, and this again just helps to hold back important things and allow water and waste substances to continue on in the urine production process. So the rate at which this happens can be measured. It's called the glomerular filtration rate, or GFR. This is something that is very characteristic of kidney function. Measuring the GFR can give an indication of whether the kidneys are functioning well, or if maybe perhaps there has been some type of damage to the kidney, and maybe they're not functioning as well as they should be anymore. So this is a useful thing to be able to measure clinically. And it's just it's a measure of the volume of filtrate that both kidneys can produce in a minute. So volume per minute. Average for a normal healthy kidney, average is about 120 milliliters per minute. That's a large volume. That's a large volume of filtrate. That's 7.5 liters per minute. So that means, think back to total blood volume. Remember, total blood volume is like 5.5 liters. So this means that all of the blood in our body is approximately being filtered every 40 minutes. So that's amazing. It's amazing how quickly the kidneys can process all of that fluid. Okay, so if the water is passing through into the filtrate, right, we don't want to just lose all that water in urine. That would be terrible. We'd become dehydrated extremely fast. So it turns out most of that water that just passed through and became filtrate, most of that water needs to be recollected. It needs to be rerouted and joined up with the blood cells again. And that's what's going to happen. We're going to learn how that takes place. Okay, so before we get into that, some things that influence the glomerular filtration rate, there are a couple of possibilities. There is actually nerve innervation that can stimulate these arterioles. Depending on the diameter of those arterioles, if they are constricted or if they are dilated, that's going to influence how quickly the blood is flowing through the glomerulus. So that in turn will influence the glomerular filtration rate. And then there's also, built within the kidneys, there's auto-regulation mechanisms in place. And this is important for helping to keep the glomerular filtration rate constant or approximately constant. So right within the kidney, what can happen is if the kidneys detect a drop in blood pressure, for example, or a rise in blood pressure. Let's go with a drop in blood pressure. That can be detected by pressure receptors, baroreceptors, in the kidney, and then the kidneys can initiate either constriction or relaxation. Constriction, if we're talking about a drop in blood pressure, the kidneys will initiate constriction of the afferent arterioles, so the ones that are coming into the glomerulus, and then that helps to keep the rate of fluid flow constant. So filtrate production stays relatively constant, even though blood pressure might have dropped for some other external reason.